So we look at the whole genome at each of those time points with our colleagues at TGen, since the MMRF are in the room with sponsorship from MMRF. And we see things that we sort of are not surprised by. So here's genome number 1. This is what she's diagnosed with. Here's genome number 2, 3, and 4. And I told you we think of cancers as a linear event, and that some initial things happen that make it cancer in the first place. So here's a mutation. This is chromosome 12 in the cancer cell. And by the way, normal would look like this. It's just straight up and down black is normal. So this is pretty messed up stuff here. Down here we see a delete. This means part of the chromosome is just missing. It's gone. That's that green spot here. And you'll see that diagnosis, first relapse, second relapse, third relapse, that never changes. So that tells us, and that's different than normal DNA, which she was born with. So that tells us that's what we call a driver event. That probably started the ball rolling. That's when the brakes failed. But here was the thing we found, and the reason I'm showing you this, which really caught us by surprise, because we always thought this was linear. One tumor led to 1.1, then 1.2, then 1.3. But in fact, if you look at what happens to Mrs. Healy here, she's got a huge chunk of chromosome 8 missing when she's diagnosed, so we treat her with Revlimid. And then we repeat her bone marrow. At a time she's taking the drug, but she's now completely resistant to it. So something changed. And in fact, we see that, in fact, that's completely gone now in the tumor. There's a totally different area of the chromosome missing. Now, you can't regrow a big chunk of DNA like that. It just doesn't happen. So what that told us, and it was somewhat surprising, is that she must have had 2 tumors present at diagnosis. And one of them went away completely with Revlimid, and another one that completely unresponsive to the drug emerged. And then a really weird thing's happened. We treat her with a different drug, and this tumor goes away, but the first one comes right back again. And that has an implication I'll explain for you in a second. What this tells us is that this patient had a change in a normal cell that led to monoclonal gammopathy of undetermined significance. She went on to get cancer. We found 5 of those driver things. There were only 5 changes. It never changed throughout the time of this patient's tumor. But those different tumor types, there was at least 2 of them. We actually know there was more than that. And one of them was just sensitive to one drug, and one was sensitive to the other. But the converse is one was resistant to one drug, and one was resistant to the other. We took one step further. We sequenced her whole genome at every time point. That's a lot of work. That's about 500 billion bases of DNA. It takes us a week on the supercomputer at Arizona State University to put one of those together. We did it 5 times. And this, to date, is the only time this has been done in myeloma ever, or any cancer for that matter. This is what it looks like if you want to see a cartoon of a genome. This patient turns out to have a break in chromosome 4 that joins chromosome 14. These are the different chromosomes, and this is that green and red stuff I was showing you earlier. And each of these changes along the outside is a mutation in the DNA. So this is kind of crazy stuff. And we saw the same thing. And the reason we did this is we want to know what those gene changes are, right down to the single DNA change that makes that resistant to one drug and sensitive to the other one. And we see here the DNA she was born with, we call it the germline. The diagnostic sample, we see a mutation in just one base of the DNA is gone, and it comes back again. But we also see what we thought we might see in the beginning, which is there's continuous mutation happening. We keep hitting new cars along the way to the end. And here it is down here. Only in the very last sample do we see this mutation. So it's continually evolving. We've done this now, you just heard, it came out 2 days ago. It's my first and only Nature paper. And this we did with a whole series of cast of characters you'll see there. We've done this analysis of the genome in myeloma in 38 different patients with colleagues at TGen and across the country, particularly at the Broad Institute in Harvard. And I'm not going to tell you the details, and this was only done at a single time point. This longitudinal study we've done is still somewhat unique. What we didn't find here, which was disappointing, We didn't find one genetic change that was shared across all samples that we went, eureka, that's the answer. We found some common changes. We found one gene mutated for which there's a drug. 8% of patients have a mutation in a gene that we have a drug off the shelf to use that we didn't know was there before we did this. So for 8% of patients, that offers some hope. Here are the implications of this story, if you think back to what I just told you. Number one, high-risk disease is very genomic. Well, number one, high-risk disease has got multiple clones present. There's not just one clone. They have different drug sensitivity. What does that tell you? Does anyone want to give you the answer? Don't be shy. One drug will not cure this disease, okay? No chance. So there are still people in the field that say, you know, it's okay to give one drug because it will work. I don't think it is okay. It means you need multiple drugs if you're ever going to get rid of myeloma. The second thing we learned is that at least in high-risk disease, that bad group of patients, the genome is profoundly unstable. Do you know what chemotherapy does? Conventional chemotherapy? It damages DNA. So this is just hypothetical, but my colleague Dr. Bergsegel is very smart, and his father, he used to love this drug called Melphalan because his dad developed it. And but you know what Melphalan does? It damages DNA. So Dr. Bergsegel, after supporting this drug for years, now turns at every meeting and says, why on earth would you give an unstable genome a drug that makes it more unstable? So that's the second implication. That's theoretical, but we might make things worse, not better. The third one, for the oncologists in the room, is that what we saw in that patient was a re-emergence of a drug-sensitive clone. And because in cancer we always thought it was a linear disease, we never pull the same drug off the shelf again. Once you become resistant, we label you as resistant forever, and we don't ever use that drug again. I think had we known in Mrs. Healy what we were going to find, we would have used Revlimid again, because that tumor went away with Revlimid. And our problem was that we never even considered using it because we thought she was completely resistant to it. So those are the three implications. I may be over my time already, but how am I doing? I might stop. Let me just go really quickly through the second part of the story. The second part of the so that's story one. Story two is how do we find a drug when we find that we found one of those Achilles heels. And I'll just quickly go through this. Well, we used robots and we used the genome again. And here's an experiment we did in TIJA. And we took a little place like this. We put some myeloma cells in each well. There's 384 wells in this plate. And at each single well, we turned off one gene in the genome. And we went through, we did this for all 8,000 genes that we had a switch we could turn them off with. And then we did it alone, and we did it in the presence of a drug, Velcade. And what we were trying to do here, we did in different concentrations of Velcade. There's the whole experiment on one plate. 150,000 experiments done all at the same time. And what we wanted to see here, this is increasing concentration of chemotherapy so that you go from no death of the cell to green, which means everything's dead. What we're looking for here are, when you turn off a gene in the cancer cell, does it make the drug work better? Do you kill it at lower concentration? That's what, if you plot out the sensitivity of the cancer cell to the drug here, this is the normal, these are all the controls in the middle. What we're looking for is something down here, which means at really low concentrations of the drug, the cells are drying much faster.